Coevolution in the Deep History of Symbiotic Fungi and Plants. Previous events create reusable modules, new opportunities, and change the probabilities of potential future interactions and consequences of those actions. When fungi and algae reached land, they both carried frameworks of previous innovations that made their subsequent symbiosis, in the context of the environmental constraints of the land, all but inevitable. Each was poised to benefit from a close association with the other, and the structures of both, particularly the vast network and rock penetration of fungi, were ideal for forming intimate associations with the roots of land plants of all kinds. The association of fungi with the roots of land plants, usually symbiotic, occasionally pathogenic, is called mycorrhizae, and they form vast symbiotic networks with local plant life. Without fungal associations, the rich evolutionary history of plants on land would have been severely limited, just as the rich evolutionary history of consumers and predator-prey food webs, complex behaviors, etc., and depend on the presence of producers. In each case, essential nutrients and energy sources are shared. Dr. Hom demonstrated how even lineages of fungi and algae that have been separate for hundreds of millions of years carry toolkits that allow them to quickly form intimate associations and share nutrients and energy in experimental laboratory encounters. There is little that is neither gradual nor random about the way that previous innovations change future probabilities. Previous innovations and complex organized entities create opportunities for higher levels of organization to arise. The rich molecular architecture of macromolecules, as well as cellular structures and developmental toolkits, all make it much easier to understand why, under the aegis of natural selection, new higher-level organizations and associations would be expected to evolve. There is no sentience or goal direction involved in this process. But in most senses of the word, these associations are not strictly random. They build on the modular, reusable framework of the past. When a previous innovation is explicitly redeployed in a new environmental context for a different purpose, we call that exaptation. But there are innumerable other evolutionary sequences that involve multiple, modular innovations that are reused in different ways. Those are more complex than exaptation, but carry the essential concept of reuse of previously evolved complex modules.